C4 plant. What exactly is a C4 plant and how we can differentiate it from C3 plants? So C4 plant or Hatch and Slack cycle was worked out in a separate group of plants like sugarcane and maize, where we found that the fixation of carbon is going to occur entirely different with respect to the C3 plants. Okay, and the genesis of this difference lies on the important thing, and that is the Crane's anatomy. So first of all, let's learn what exactly is a Crane's anatomy. Have a look at this diagram. So this is the anatomical structure of the leaf, you can see there, right? So this is a bundle seed, right? And this is a mesophyll. Okay, so we say there are dimorphic chloroplast or dimorphism. What is the meaning of dimorphism? That there are two different types of chloroplast, the bundle seed chloroplast and the mesophyll chloroplast. Both chloroplast has been assigned a different role. In a very simplistic manner, if I say that bundle seed chloroplasts are primarily aimed for performing the Calvin cycle, and mesophyll cells are primarily concerned with the performing the light reaction job. Okay, so that's why there is somewhat differentiation in the job, and thereby making it more and more efficient. Okay, so this is what we call Hatch and Slack cycle. Now have a look at the entire cycle. So as I told you, there are two things. One is the mesophyll, second is the bundle seed cell, right? So in case of mesophyll, what we see that the first CO2 acceptor is going to act as a PEP. Now it is different from C3 cycle. In case of C3 cycle, it was a 5 carbon compound RUBP, but here the phosphoenol pyruvate, that is a 3 carbon compound that is going to act as a CO2 acceptor. Okay, so this accepts CO2, and as a result what we see, oxaloacetic acid is going to form. This is a 4 carbon compound, the first stable product, and that is why this entire cycle is actually called as a C4 cycle. Now this oxaloacetic acid gets converted into malic acid, okay, and the malic acid is actually being transferred to the bundle seed cell, right? Now in the bundle seed cell, what happens, this malate undergo the decarboxylation process. That means there is a 4 carbon compound, one CO2 will come out, right, and one CO2 will come out. As a result, the 3 carbon compound pyruvate is actually being formed. Now this single CO2 is being accepted by RUBP, right, and within the bundle seed cell, what happens, that Calvin cycle will run and eventually form the starch. Now this pyruvate will again go back to the mesophyll cell. In the mesophyll cell, what will happen, the pyruvate will undergoes dikinase activity, and as a result what will happen, that 2 molecules of AT being utilized, and again the CO2 acceptor in the form of PEP is actually formed. Okay, so this is how the entire Calvin cycle is being completed. This is how entire Hatch and Slack cycle is actually being completed. Now mind it, the passage of molecules from mesophyll to bundle cell is restricted to few molecules only. And what are they? The very first, the malate, the second, the pyruvate, okay, and there is one more, what we call NADPH2, because you know in Calvin cycle NADPH2 is required, and this NADPH2 is actually being formed in the mesophyll cell only. So once the mesophyll NADPH2 is being formed, these NADPH2 goes to the bundle seed cell, wherein the Calvin cycle is actually being utilized. But in addition to that, there is one more molecule where the passage from mesophyll to bundle seed cell can happen, and that is in the form of aspartate. So there are few plants in which instead of malate, the oxaloacetic acid gets converted into aspartate. So aspartate gets transferred to the bundle seed cell, and here again it gets converted into malate, okay, and then rest of the reaction remains the same. So again malate will be decarboxylated, CO2 will be utilized by the Calvin cycle, pyruvate will be formed, pyruvate will again go back to the mesophyll cell, where again it will regenerate the RUBP, it will regenerate the PEP, that is the CO2 acceptor. Okay, so this is all about the aspects of the Hatch and Slack cycle, or what we call C4 cycle. Okay. Now let's talk about the energetics. Now have a look at this. Here what happens, 5 ATP is required, okay, and 2 NADPH2. So 5 ATP and 2 NADP is required for the formation of 1 molecule of carbon dioxide. How the 5 ATP? Because you see 3 ATP was required in Calvin cycle and 2 activity was required in dikinase. So 3 plus 2 makes 5. And as far as NADH2 requirement is concerned, that remains the same. So in Calvin cycle, the 2 NADPH2 is required. So here also it is the same. Okay. So 1 molecule of carbohydrate require altogether we can say 30 ATP and 12 NADPH2. Okay. This is the most efficient mechanism for the CO2 fixation, or rather it is more efficient than C3 cycle. Now why it is more efficient? The reason behind their efficiency lies in the Kranz anatomy. What happens that the Kranz anatomy ensures the complete carboxylation of within the bundle seed cells. There is no oxygen, right? So it ensures the carboxylase activity, thereby more and more fixation, okay, and more carbohydrate being formed. The Kranz anatomy is primarily responsible for making it more efficient, and due to Kranz anatomy what happens, the photorespiration is not there. Okay, what exactly is the photorespiration? We will discuss about in the next session. So that's all as far as the Hatch and Slack cycle is concerned. Thank you.